All right, we are wrapping up the digestive system by talking about the accessory organs, which are the liver, pancreas, and gallbladder. So, accessory organs, again, are those that are outside of the immediate GI tract, so they're not part of that big hollow tube, but they are really, really important for chemical digestion in the small intestines specifically. So, the accessory organs are all located very close to the early on parts of the digestive system. So we've got our liver up here, gallbladder underneath it, and then pancreas kind of sits around the bottom part of the stomach and upper portion of the intestines towards the back of the body, so towards the posterior side of the peritoneal cavity. So the liver has four lobes. There's two primary lobes on the anterior side, right and left, and then the two smaller lobes are the caudate and quadrate, which are posterior. And there are 5 different ligaments or peritoneal folds in the liver. We learned the falciform ligament in lab last week, which is one of the 2 remnants of the umbilical vein and separates the right and left lobes from each other. In conjunction with the ligamentum teres hepatis. And then there's also the coronary ligament and 2 lateral ligaments. And they're all just going to help make sure that the liver is staying in place, just like all of our other mesenteries are making sure that the organs stay where they should be. So within the liver, we have what are called hepatic lobules. These are actually in a hexagon shape. They have a central vein down the middle, which is taking deoxygenated blood away from the liver. And then we have the portal triad at the corners, which have 3 structures. And one of those is the bile duct. So the hepatocytes, the cells making bile, are going to carry those. Then we have a branch of the hepatic vein or portal venule carrying deoxygenated blood rich in nutrients from the digestive tract. So that's the hepatic portal vein. And then there's a branch of the hepatic artery, which is going to carry oxygenated blood to the cells of the liver. So the direction of blood flow, again, this is a refresher from lab. Hepatic portal vein is bringing deoxygenated blood that is rich in nutrients from the digestive system into the liver. The hepatic artery is also going to bring oxygenated blood into the liver to the hepatocytes, and then we are going to take that to that central vein. Bile is going to flow from the liver cells to the bile canaliculi, which are little openings, and then that's going to take it to the bile duct in the portal triad. So basically, blood and bile are flowing in opposite directions from each other. So bile is made by hepatocytes or liver cells. We make about 1 liter a day, and this is a mixture of water, bile salts, bile pigments like bilirubin, which is green. So this is one of the pigments that is present during the breakdown and repair of bruised tissue. So that's why bruises will sometimes turn that greenish color, is that bilirubin. And then there's also phospholipids, electrolytes, cholesterol, and triglycerides to make up bile. And its job, again, is to emulsify or break down lipids in the small intestine. So here's our image. So here is those lobules with that hexagon structure. Here's a zoomed in view. So we've got the hexagon, we've got that central vein in the middle, and then there's all these other veins on the outside that are coming from that hepatic portal vein. And then we also have that hepatic artery, some branches off of that, going into all of these little capillaries, or sinusoids is the official name for them here, which are taking blood to the central vein, and then that will go out the hepatic vein to the inferior vena cava. We've got those bile ducts here, so the cells inside of the liver, those hepatocytes, are producing the bile and secreting it out into these bile ducts. Next up is the pancreas. So the pancreas is a glandular organ. It is retroperitoneal, so it is outside of the peritoneal cavity, and it is posterior to that peritoneal cavity. It is both endocrine and exocrine functions. So endocrine for hormones like insulin and glucagon, and then the exocrine function makes digestive enzymes. So it has a head, body, and tail, as well as the pancreatic ducts that we covered in lab last week. So the head is right next to the duodenum, and then the body is going to be most of it, and the tail is the very end closest to the spleen. So the pancreas has a lot of different types of cells, but here's a couple key ones. The islet of Langerhans cells. These make the hormones. So this is going to make the insulin and stuff like that. So this includes the alpha and beta cells. And then we have acinar cells, which make the pancreatic juices containing all of the different enzymes and stuff that we want going to the small intestines. So the pancreatic juice is made up of salts, the sodium bicarbonate, baking soda essentially, and then a bunch of different digestive enzymes for carbohydrates, proteins, and fats. Enters that duodenum by the hepatopancreatic ampulla, or the sphincter down there. The secretion is controlled by hormone function and the parasympathetic nervous system, so that rest and digest response. So here are just some graphics. So we've got our common bile duct here. Here's those pancreatic ducts, again, going into the duodenum. Tail is out here. And so these lobules are going to look like this when we kind of zoom in. So we've got our acinar cells making the digestive enzymes, and then the Langerhans cells, the pancreatic islets, for the hormone production. So our pancreas has a couple different really important functions. And so again, we need that bicarbonate for the neutralization of hydrochloric acid coming from the stomach. It's going to also inactivate pepsin coming in from the stomach, and so that is going to allow better environment for very pH-sensitive digestive enzymes to start working in the small intestine, and so those are going to help break down more fats, more sugars, and more proteins than we have earlier on in the digestive process. So the pancreas enzymes are actually produced in their inactive forms, and then they get activated in the actual duodenum. And the reason for this is if they were produced in their active form, they would actually digest the pancreas, which we obviously don't want. This is exactly what happens in a disease called pancreatitis. So that's going to cause a lot of inflammation and breakdown of the pancreas cells. So that's not good. And the reason that we do not have that happen in the small intestine is the cells in the small intestine are going to have that mucus layer over them again, and their job is to do a lot of absorption. So between the mucus and the absorptive properties, we are not going to be digesting the small intestine. So in the intestine, the brush border, those microvilli are producing enteropeptidase, and that is going to trigger the activation of all of those different pancreatic enzymes for the digestion in the small intestine. And last but not least is the gallbladder. So this is going to be on the underside of the liver. This is a muscular sac. It is made up of a few different regions, the fundus, body, and neck. It is going to store, concentrate, and then release bile into the duodenum. Its mucosa is made out of simple columnar epithelial cells with rugae, just like the stomach. There is no submucosal layer in the gallbladder. And then we have these smooth muscle layers to actually expel the bile from the gallbladder. And we've got part of that visceral peritoneum helping to hold it under the liver. And again, you can live without a gallbladder. It can be a lot harder to actually break down some lipids, though. So bile is going to start again in the liver with those hepatocytes. Then they travel through the bile duct in the portal triad to the right or left hepatic bile ducts. And then the common hepatic duct, then to the cystic duct, if we're going to store that bile in the gallbladder for a little bit, or directly to the common bile duct, so it can leave to the duodenum via that same hepatopancreatic ampulla or sphincter. And so it can take a couple different directions depending on what's needed at that particular point in time. If we just ate a very, very fatty meal, it's probably just going to go straight to that common bile duct. If we've not eaten recently, then that bile would end up getting stored in the gallbladder. So here's that gallbladder. Again, we've got the rugae inside. Here's our cystic duct right there, our right and left hepatic ducts. And then we've got our common hepatic and then common bile duct. So lots of tubes for moving bile around depending on where it actually needs to go. And that wraps up the digestive system, and we will continue in the next series with the respiratory system.